All right, hey everybody, welcome to episode 17 of Dr. D's Discussions. I'm a little upset right now because my computer's having some technical problems and my microphone has some feedback issues. So if anybody knows how to fix some feedback issues on a microphone for a Windows 10 device, please let me know. I'll put out a Facebook message later tonight and see if anybody knows how to do that. Otherwise, I'll have to rely on Jefferson Technical Assistance, and I don't think they're really going to be able to help me. But we'll see how it goes. So, are we having the same issue?
Keep talking.
Yeah. Oh, all right. So tonight's episode is on the eukaryotic cell, and we're going to be breaking it down. What a eukaryotic cell is? It's the plant, the animal, fungus, the fungus among us, protists, amoebas. And Rosa locked herself out of the room. Jeez, man, we're on a roll tonight. Yo, we are like batting 1,000 tonight. Jeez, it's all good though. That's why we like to have a little fun around here and keep it light. So we'll keep it light. Rosa, just look up the times that IT is open tomorrow for me, so I can get on that. All right. So, Lee, what's your favorite cell?
Natural killer cell.
Nice, all right. I would have to say my favorite cell is, I don't know, maybe a muscle cell, because I'm so swole. But, all right, so basically what we'll give you the rundown. We're going to give you an overview of what a eukaryotic cell is. We're going to give the endosymbiotic theory of the evolution of the eukaryotic cell from prokaryotic origins, and then we're going to go into what the organelles of the cell are and what they each do in terms of their job. Then we're also going to detail about what I did in terms of research for my master's thesis, and maybe we'll do for my PhD thesis. And we'll definitely get into some of the topics that you'd like to investigate in the eukaryotic cell. So
yeah,
you
we'll talk some DNA stuff. We'll talk some regulation stuff.
Yeah, okay. We'll talk some stuff.
Some
things and stuff.
Some stuff, you know.
Things and stuff. All right, cool. All right, so let's get to it. Here's a rough overview of the eukaryotic cell displayed behind me. If you can see that, hopefully you can. If not, we'll break it down for you. It's a plant cell, so it's got a bunch of stuff and things. So you got your nucleus, which contains your DNA and all your genetic material. Also has some proteins, ribosomes, and fun stuff like that. You got your nuclear envelope, your nucleolus. Then outside of your nucleus, you have the ribosomes. Around your rough endoplasmic reticulum, you have your smooth ER, or your smooth endoplasmic reticulum.
Pretty much these are places where protein folding and packaging occur. Mm-hmm. Once mRNA leaves the nucleus and gets translated.
Okay, yeah, we'll go into a little bit of detail about that as well. We'll talk about the Golgi Apparatus, or the Golgi body. And that's where basically proteins get modified with carbohydrates and different things like that. Also post-translational things in terms of like phosphorylation and other aspects of that, like glycosylation. What else is there? What other ations are there? Polyadenylation. There's a long list.
There's a lot of them.
Yeah,
this is like there's a whole field of biochemistry devoted to post-translational modifications.
Nice. And then we'll talk about the mitochondrion, which everybody knows is
Powerhouse of the cell.
Yep.
Go ahead, do it, Dave. Do it, part.
Woo! Woo! Yeah, powerhouse. All right. In plant cells, you actually have what's called a central vacuole, which is filled with water, I believe. I'm not very familiar about plant cell biology,
I got
so forgive me on that.
I got some plant biologists on my Facebook. Please chime in if we get anything incorrect, because we are not
plant biologists at all. So
I'll keep it light with the plant stuff.
Yeah, we'll have to keep it real light with the plant stuff. Oh, jeez. Then we have what's called the intermediate filaments and microfilaments, or the actin-based filaments, and those are basically the structural support of the cell. And basically, what that does is literally.
Well, they give they allow cells to. Well, in plants they don't.
They don't really
plants
move.
have cell walls, so they can move. But in normal or in other eukaryotic cells that just have membranes, that's what allows a cell to migrate and traverse through whatever medium it's in, whether it be water or blood or whatever the case. That's pretty much cell movement is mediated by these cytoskeleton proteins.
Yeah, so yeah, that's a great explanation. Damn, couldn't say it better myself. Yeah, so let's pop to it. So Lee, endosymbiotic theory of evolution. You go with an ancestral prokaryote, which is either your bacteria, archaea, and those basically mean simple cells in terms of a cell biologist.
Single-celled organisms that lack nucleus. Pretty much, they're a giant. They're essentially a bubble with proteins and stuff inside. Yeah, like all their information is pretty much floating around. I mean, it's localized to areas within a bacterial cell, but there's no real barriers or compartmentalization associated with prokaryotic cells. That's one of the major things that separates prokaryotes from eukaryotic cells is that the ability for eukaryotic cells to create compartments within itself. And inside of the cell, you can modulate pH and concentration gradients because of this compartmentalization.
And basically the reason why this compartmentalization occurred is to be through the infolding of the plasma membrane into the interior of the cell. So you had this infolding occur early on, and it basically surrounded the nucleus and created a nuclear envelope so that you can sequester your chromosomes or your DNA into one area so that you can protect it from radioactive species, reactive oxidative species and other different things like that. And then you had more infolding and more infolding and more infolding to make the ER and different things like that so that you sequestered protein making abilities into one area. And then you come across you're this cell with all this infolding and you're like big, and then you're like, wow, I can't get energy where I need to get energy. So you engulf a small little aerobic bacteria heterotrophic prokaryote.
Yeah, so likely, so the first, from an evolutionary perspective, glycolysis was probably the first metabolic pathway ever. Since it's in every cell, every single living thing on this planet does glycolysis at some level. Eukaryotic cells figured out a way to maximize energy production by pretty much piggybacking some of these prokaryotes. And then we have the endosymbiosis theory is pretty much coevolution between a primitive eukaryotic cell and a prokaryotic cell, kind of just working together.
So this little aerobic heterotrophic prokaryote had figured out how to use oxidative phosphorylation by some means, and it was just making massive amounts of energy. But at the same time, it wasn't necessarily the greatest at engulfing new materials.
Right. And there's precursors involved in making energy.
Yeah. So, like glucose.
Prokaryotes, more specifically.
Or acetyl-CoA.
Yeah.
And then that gets to the point into the Kreb cycle, and the Kreb cycle, the citric acid cycle, spits out all these nice, fun molecules in terms of getting through the electron transport chain. And so these two things work together to essentially make what's now called a eukaryotic cell.
Right.
I know that it's kind of like the analogy would be how we have bacteria in our gut, and we kind of rely on them for their metabolites, and they rely on us to ingest food and different things like that. So it's basically
it's another form of a symbiotic relationship. I mean, in that sense, it's more of a macro scale, right? We are this giant organism, and then we kind of have miniature organisms inside us. But when you look at the cell, there's a bigger organism, so to speak, with a miniature organism living inside of it.
Yeah.
It's a very similar, association as far as the demands and the relationship is concerned.
Yeah. So I think that's pretty interesting little theory of evolution that we could throw into.
Yeah, I mean, it's got some data. It's definitely got some data that supports it. I mean, mitochondria and chloroplasts still have
their own little
genome.
DNA.
Yeah,
they have their own DNA. So yeah, that's also one thing. Also, the lipid membrane layer for both chloroplasts and mitochondria more resemble those of bacteria rather than what our cells are, eukaryotic cells. So I think that's also another really interesting point. So other than that, I think. Do you have any other points?
Not
other
for this.
points?
Not for this theory. No, not for this theory.
All right, cool. All right, so. Let's give a little breakdown of the organelles and their function, and then we'll go into a little bit more detail with the pictures and talk about it a little more.
So just to, not a disclaimer, but just to set the premise of this next slide is that prokaryotes don't have organelles, right? And they don't have organelles because they don't have the ability to create compartments. And
By the
so,
unfolding of plasma.
right, and so
the
the ability to create, compartmentalization kind of led to the ability to form organelles, and that's how that occurred evolutionarily speaking.
Yeah,
But
it's more specialization. So think of it this way, when we first started off as primitive people in the, plains of Africa, we're all hunting and gathering, and there was some specialization. Then we started settling down, and one person became a farmer, one person became a carpenter, one person became this and this and that and the other thing.
Right.
So
imagine they're all working as a giant organism.
Yeah.
It's kind of
civilization is just one big giant organism, and we're all specialized to do certain jobs. We're all basically cogs in the system. So think of each organelle as an analogy, rough analogy, as a person doing particular jobs so that the whole can actually function as it's supposed to. Nucleus, the organelle that contains the cell's DNA, is the control center of the DNA, or the cell, rather, DNA. Ribosome, the organelle in which amino acids are hooked together to make proteins. That's a pretty simplistic way of looking at it, especially I know with Dr. Ho's lectures coming up for us, we're going to have to know the ribosome in and out, and left and right, and sideways, and up and down, and the S30 subunits and the S70 subunits.
Super complicated stuff. Yeah. I mean, ribonucleic proteins and, with
tRNAs and
ribosomes and RNA modifications, yeah. Anyhow.
Endoplasmic reticulum, the organelle that makes lipids, breaks down drugs and other substances, and packages proteins for the Golgi Complex. Golgi Complex, the organelle that processes and transports proteins and other materials out of the cell. Like we had said, it actually adds different molecules onto proteins and different things like that. Mitochondria breaks down food molecules to make ATP, powerhouse of the cell. Woo! Lysosome, the organelle that digests food particle, waste, cell parts, and foreign invaders. Central vacuole, the organelle that stores water and other materials.
Right. I want to talk about the lysosome real quick because the lysosome is an interesting organelle, man. Essentially, it's toxic.
Yeah.
It's
got a pH of what? 5?
Less, I think. I think
3
it's
to 5?
Actually, let's get some Googlers going real quick.
Yeah, Google on that.
Yeah.
Nice.
Let's get some verification on the lysosome pH.
4.5 to 5.
Yeah. Well, you were pretty spot on with that one.
Yeah, I thought it was pretty good.
I thought it would be lower than that, given that
the stomach is at 3, so
right.
I think it's
maybe that's what I was thinking of.
Maybe.
But essentially, though, I mean, most things don't function at the pH of 5, and if lysosomes started to just pop all over the inside of the cell, pretty much the cell would die. One of the ways that cells recycle things is that they fuse other organelles to lysosomes so that the contents can be degraded.
autophagy.
Yeah.
Yeah. Nice.
Essentially, yeah.
And there's specialized enzymes that work at subphysiological pH, such as 4.5 to 5, that are only activated in that pH and in that environment. So going back to the compartmentalization and basically how that works hand in hand with the concept we just said is that, let's say these enzymes got out into the regular portion of the cell, they're not going to break down any proteins or other things or the other organelles because they can't function at a higher pH, whether that's through folding or chemical reactions or whatever. Chloroplast, the organelle that uses the energy of sunlight to make food. I like food.
Primary producers, man. Good old plants.
Gotta love plants. It's food that food eats.
Food that food eats. Right. Like cows and chickens. Shout out to all my vegans out there.
All right. So let's talk about the nucleus, the nuclear envelope, and the nucleolus.
It's my favorite part of the cell.
Yeah, that's your baby?
That's my baby right there.
All right. Well,
So
I'll let you take the lead on your baby.
That's where all the magic happens, The rest of the cell is just kind of there. It's not
as a tyrosine kinase guy and a protein pathway guy,
I'm
a little angry.
I like the cyclic transduction stuff, but the cell goes nowhere without the genetic information.
Yeah, that's true, but without any outside information from the extracellular regions, you're not doing nothing with that nucleus. Why, you can just ignore the outside world and just do your own thing?
So I'm gonna say the disclaimer here is that me and Dave are both, you guys know our educational studies and stuff, but we study two different parts of the cell.
Yeah.
So our interests lie in different aspects. But as far as the nucleus is concerned, things move in and out of the nucleus. DNA doesn't, for the most part, unless it's virally mediated. But just for conversation's sake, all of the genetic information is contained within the nucleus. There are proteins inside of the nucleus that operate and pretty much manage your genetic information and regulate when it gets, accessed and repressed or whatever the case may be. And when your cell gets ready to divide into another cell, a bunch of different things have to occur to the nucleus, such as breaking down this nuclear envelope and then rebuilding it back together again, so that all your genetic information is maintained. And the nuclear pores facilitate what kind of gets in and out of the nucleus, and it's analogous to like the blood-brain barrier.
Nice. That's a good analogy,
everything doesn't come into the brain for obvious reasons. It's super important. And so the same thing occurs with the nucleus. You don't want certain proteins that don't necessarily have a function there going into the nucleus. And as you guys can see, there's this theme associated with the ability to create compartments and regulate those compartments.
Yeah, that's very true. And if you don't, and there actually is a new theory, I don't think it's new, but subcompartmentalization, supported by the data that is seen, subcompartmentalization of the nucleus, rather. So that's pretty interesting.
Yeah, that's an interesting thing that has. So speaking of the nuclear pore, there's some report. I want to say some report. There's plenty of literature out there that suggests that the interior of the nuclear pore has a lot to do with genomic information regulation by being able to localize areas of heterochromatin and then having euchromatin not necessarily patched against the nuclear membrane, which allows it to be transcribed. There's a couple of theories out there as far as the genetic information gets regulated at a three-dimensional architecture perspective, which I kind of will get into later at the end of the show.
Cool, perfect. All right, prokaryotes versus eukaryotes in terms of genetics. Prokaryotics are often haploid. Eukaryotes are often diploid. So basically that means that you have one copy of one gene and prokaryotes, and in eukaryotes you have two alleles, or two copies of the gene.
Or sometimes three if you're a little Down syndrome that day.
Oh, Jesus. A little polyploidy?
Yeah, a little polyploidy. Although I will say this, though. Polyploidy in humans is usually lethal or extremely debilitating.
However,
but in plants, it is very common.
Especially in sunflowers.
Oh, really? Yeah.
Sunflowers can have like an N of 8, 16, 4, 2.
And when we say N, we don't mean statistics. N stands for sets of
chromosomes. So everybody has, 23 pairs of chromosomes, 22 autosomes, one sex chromosome.
One set of sex chromosomes. You have two sex chromosomes,
those are usually come in pairs, and that's what you get diploid.
Right.
If you double that, you have quadruploidy.
Right.
Or tetraploidy, rather, sorry.
Yeah, quadruploidy.
Yeah,
somebody.
Making some words up over here. But basically, in plants, you can have up to 16 times of the chromosomes. So then you have, I don't even know what that would be. 16
I don't know what a 16. Yeah, somebody, what's like the 6 for 16ums? I don't really know. But it's an interesting fact. A lot of times people get online and talk about how like seedless fruit is like genetically modified, but it's not. Actually, if you just double up or triple up the amount of sets of chromosomes inside of a plant, you can get part of its like reproduction set to be seedless.
Nice.
Yeah, the watermelons and grapes, it comes from like having extra sets of chromosomes. They're just seedless by nature.
And then a lot of those are actually replanted based upon just taking like a sprig or like.
Well, it depends on certain
certain
fruits and. I'm not sure.
When you actually just take like a little piece
so
of
you can take like leaves and stuff like
and
but
grow a new tree.
Yeah, I think it's like plant dependent.
Yeah.
Again, I don't really know.
We're not plant geneticists.
I don't really know, but I'm pretty sure. I've heard of that before. I don't really know if that's some folklore or if that's actually real biology because I have about. This is my education. This much is plant biology. And I was just like that 1 week in Biochem 2 where we had to learn about different aspects of photosynthesis and C4 cactus plants produce energy at night and stuff like that. Yeah, it was just a weird. Shout out to plant biologists. We need you guys.
Yeah, thank you. We need you in agriculture, especially with climate change coming up.
Yes.
But let's also go into it. Prokaryotes often contain plasmids or circular DNA that's just floating around in conjunction to their chromosomes, their singular circular chromosome. Eukaryotes have linear chromosomes and usually more than one, I would hope. Prokaryotes, translation is coupled to transcription. So that basically means anytime you read DNA, make RNA from it, translation occurs right away because there's, again, no compartmentalization in the cell. In eukaryotes, transcription of genes in RNA occurs in the nucleus, and translation of that RNA into protein occurs in the cytoplasm. Basically going from the nucleus through the nuclear pore, into the endoplasmic reticulum, hits a ribosome, boom.
Right.
Gets made some protein.
Yeah, it hits, the ribosome subunits and
yeah.
It's actually kind of complicated process, but
it's a very complicated.
For all intents and purposes.
What are the subunits again?
The 30S and the 50S, which make a 70S. Yeah, I know 30 plus 50 equals 80, but it's based on ultracentrifugation and not actual addition. It's a weird thing.
Yeah. Gotta love it.
But yeah, so I also wanted to talk about the whole plasmids things you mentioned.
Mm-hmm.
So in eukaryotic cells, viruses can create episomes, which are circular pieces of DNA that kind of chill in the nucleus. There aren't episomes normally in eukaryotic cells. This is like a virally mediated incidence, but I always thought it was interesting. It's actually not. I think hepatitis C actually works by this episomal latency, so to speak. It's not hepatitis. The virus isn't actually incorporated into the genome. And a few years back, these guys that produce Vivaldi found out a way to modulate that or get that broken down, to get the cell to be able to break that episomal DNA down and cure people with hepatitis C.
No, that's how they cure it.
Yeah.
I didn't know that. That's a very
because you have to get rid of that episomal DNA because it's not actually incorporated into the genome. But yeah, we should do a show on that mechanism because it's actually super cool.
It's cool. Yeah, we'll have to do that. All right, so let's move forward a little bit because I know we want to talk about our own designated research.
Our own sexy stuff.
Yeah, so we're going to give a real brief breakdown of each of the organelles. We're not going to go into too much detail. We'll basically read off the slides. Endoplasmic reticulum. Make and package proteins and lipids. Lipids are fats. Making it much like an assembly line. Interconnected network of flattened membrane-enclosed sacs. Tubes known as cisternae. Largest organelle in the eukaryotic cell. It provides separate chemical environment which allows for correct protein folding. Again, going back to the compartmentalization thing, that is so important for the eukaryotic cell. Without the compartmentalization, you can't do specialized functions.
Yep.
It's like trying to make glass in a blacksmith smithy. You're not going to be able to make glass in some place that you're making a goddamn sword. So, you know what I mean? Then, it's compartmentalization. Or you're not going to be able to read a book inside of an airport. You know what I mean? You have to separate these things out.
Right.
And then there's rough endoplasmic reticulum, which means that there's ribosomes stuck to it, or smooth endoplasmic reticulum. Now, you might have to forgive me because I forget what the smooth does over the...
I think the smooth is more about...
lipid, synthesis, right?
Yeah, I don't know. And we should know because we're biologists. But
cell biologists.
I'm pretty sure it's the smooth ER is associated with transport, but that may be. Let me check my notes real quick before we get into trouble here.
Get into trouble? Oh no, we're screwed.
Oh, no. Yeah, you're right. They synthesize lipids and steroids.
Thought so. Well, I'm not too degenerate.
They're also associated with some metabolism. They connect to the nuclear envelope. Yeah, they do stuff like that.
Cool. Ribosomes.
They make proteins.
They take mRNA and they use that codon sequence, sequences of the nucleotides per codon. A codon is 3 nucleotides that gives TAG or GAC or whatever to give you a specific amino acid.
CCA for tryptophan or something like that.
CCA for tryptophan? I don't know.
Yeah, I
UGA is the stop codon. I know that. University of Georgia.
Yeah. AUG is the start codon.
Yep. And then, what is it? UGG?
Yeah. UGAG, man.
UGAG.
Yeah.
UGAG. I love it. It's a little acronym that you use to
also,
memorize it.
It's like baby talk. I don't know. It's weird.
UGAG.
But yeah, we had to memorize the stop codons like 15 times during biology education. It's horrible.
Yeah. And then I'm so glad I don't have to memorize amino acids again.
But yeah.
The codon per...
So pretty much the way that ribosomes are pretty much like a sandwich. You got a lower part and the top part. And the lettuce and tomatoes is the mRNA. The lettuce and tomatoes is the mRNA. So when you get the sandwich together, one of the byproducts of that is actual amino acid chains that end up folding and creating proteins. And this creation of these amino acid chains is actually mediated by the top part of the burger, in this case, which would be the large ribosomal subunit.
Yeah. The 80S is the total, and
Right.
then the 40 and 60.
Is it 40 and 60?
Yeah,
Are you
the
sure?
No, 30 and 50 is the
Prokaryotic. Okay, I knew
Yeah.
those numbers were wrong.
Yeah.
Yes.
So I was like, wait, I don't want to be wrong about this, so I googled it.
Right. Prokaryotic is 30, 50, 70.
And basically, the mRNA is recognized in terms of its Kozak sequence, which I forgot what was in class on last week.
I know in prokaryotes it's the Shine-Dalgona sequence that
Yep.
mediates.
Shine-Dalgona, yep. And then have Kozak in the
Shout out to that dude that discovered that.
Nice.
Yeah.
Shine-Dalgona.
Who's probably deceased by now, but I mean.
So basically, the ribosomal subunit, or the ribosome, reads the mRNA and then brings in tRNA that's attached to the amino acid of like choice. So, and that's how the codon sequence works. So you have 3 letters. Let's say AAA, and that
Right.
means you get I forget actually what does AA mean.
I don't know, but so you have, for each set of codons, you have a corresponding tRNA molecule
that
recognizes that codon.
and has on its butt end the amino acid.
Right.
And then it takes it, displaces it onto the growing polypeptide chain that makes a protein, and then boom, you make a protein.
Yeah.
It's pretty nice. It's interesting. We'll be going into a lot of detail with that in the next week, so
Yeah.
we'll be talking about that in our genetic information
I love
transfer
protein
class.
biochemistry.
I hate structure. Oh my god. We'll go into that some other time. Golgi body apparatus. Basically, it's where I think of it as the post office of the cell. So it kind of delivers where the proteins and all the functional things that you make in the ER to the places that need to go. Or, FedEx or UPS, whatever your preference of choice is. But it also does modifications as well. So it does glycosylation, adding carbohydrates onto different things, adding fats onto things. It really does what it has to do in terms of modifying the proteins for their specific functions. And, do you have any?
Yeah, no, you hit the nail on the head as far as the Golgi apparatus is concerned.
It's got a cis phase, a trans phase. You go from cis to trans, and then from the trans you can go to exosomes, extracellular vesicles.
Right,
Actually,
yeah, 'cause
exosomes
exosomes
go
are produced from
multivesicular bodies.
There you go.
Go into lysosomes, and then basically, you either spit the contents out into the extracellular matrix or extracellular space, or
Or
it
you
gets
use
used
the other
elsewhere.
stuff, yeah, and inside us all.
Mm-hmm. Or, if it's a transmembrane protein, it gets stuck in the plasma membrane, it stays there. And it's all, what is it, COP2, COP1, clathrin? Or
I know these interactions are mediated by Rab
RAG proteins, yeah.
Yeah.
And basically, here's a little fun fact for you. You can tell what compartment or what area of the cell
Something it's
basically
going to, yeah.
staining for Rab protein. It's very specific, like Rab, remember all the endosomes?
I know the CC45 that goes to I don't know.
Rab2, Rab3,
My brain is
Rab5 goes this way.
My brain is hurting as far as this part of the cell biology is concerned.
But, you can stain a cell based upon the Rab proteins and tell what you're looking at. And that's nice.
Yeah, the takeaway from this is that the Golgi apparatus is used for vesicular transport and pretty much anywhere around the cell.
Yeah, think of it as the post office.
Yeah, it's a great analogy.
All right, mitochondria. Again,
Powerhouse
of the cell. Everybody, I love those memes that go around Facebook. They're like, "What did school teach me?"
Yeah, mitochondria, powerhouse of the cell.
Exactly. Didn't teach me my taxes. They didn't teach me how to
right.
build something or nothing of real use, but mitochondria,
Powerhouse of the cell.
I think we should make a T-shirt.
There are T-shirts actually that say that already. Yeah.
Bio is life media.
There, we cut it. Put that on the back there. Mitochondria are also associated with cell death. And there's also some diseases associated with mitochondrial dysfunction. They make energy, and if they don't work, you die.
Yeah. And also it has you die. Jeez.
Yeah, you die.
Mitophagy.
Yeah.
So if
they do
you
water recycling of them. And the 3 parent babies started with mitochondrial deficiencies. That's where we got the idea to start making those kids from.
Yeah, basically, and that's episode 3 of Dr. Jesus.
Somebody verify that on Google, please. What?
Yeah, we might need you to help me out with that one.
Leave
But
that in the comments,
we did do an episode on basically Mitochondria have their own genomes, basically. It's very small, it's very compact. It's only, like a couple thousand base pairs. But they make 2 ribosomal proteins, and they make all their own tRNAs, or a lot of their own tRNAs. But they use a lot of nuclear-based genetics to make their proteins and make their membrane proteins and different things like that. But at the same flip side to it...
So it's 16,000 base pairs.
16,000.
Yeah. And it only contains 37 genes.
Nice.
Yeah, it's not a lot.
It's not big.
It's really small.
But if you have a mutation in those genes, you will get a mitochondrial disorder.
Yeah.
And basically, if you, like with the 3-parent child, you take the mitochondria of a...
Healthy person.
Take the nucleus out of your mom's eggs, which have defective mitochondria, and you put them into a donor egg with no nucleus in it, but all the good mitochondria.
Good mitochondria, yeah.
And basically, when you do take the nucleus out of your mom's egg, the primary mom, that you will get some mitochondrial spillover. So it's not perfect.
It doesn't matter because mitochondria, the mitochondria that you inherit from at that stage of development will repopulate and divide to such an extent. So you got to think of it as this, if you have 3 out of 100, right, and 97% healthy, 3% bad, and as that explodes into these astronomical numbers, that kind of gets diluted out.
It's a threshold too, it's a threshold disease. And a lot of these things, you need to have at least 30 to 40% bad mitochondria to see any type of defective
Essentially, all diseases are threshold mediated. You make... a lot of the times, it's not whether you have something. Right, it's not whether you have something or not. It's how much of it do you have. Of course, if you're 0, you're definitely going to get the goddamn disease. But if you need 65% effectiveness and you're bad with 43, you got to get the disease, and it's kind of crazy. But you can if you need 65 and you're at 65.5, but normal is 86, you probably won't get the disease phenotype. It's a very threshold mediated thing associated with getting a disease.
It's interesting. I really find that really interesting. And then I know we harp on it all the time, but it does have the electron transport chain and all those nice complexes.
Kreb Cycle. Did you learn that in 4th grade?
Yeah, mitochondria is the powerhouse of the cell.
Powerhouse of the cell. It's actually super complicated too.
It is. But I think it's a nice little organelle. I think it's
it's all
Nice.
right. I like it.
I don't want to study it.
Yeah, we have a whole department at this university called Mitocare that studies mitochondrial diseases and mitochondrial function.
The Al-Nemri lab studies it as well in terms of
caspase. They study apoptosis, which mitochondria is mediated. Pretty much, you poke a hole in this goddamn thing and you die.
Yeah, you're screwed. All right. Chloroplasts. It couples the use of light to the Calvin cycle. Something that I haven't looked at in 10 years.
All right, so pretty much, I am going to let Dave talk about the Calvin cycle part of this. And I'm going to talk about the light part of this because it's been a long time. So pretty much you have chlorophyll, which is a big molecule, and that has a metal in the middle of it, essentially. I think it's magnesium, or manganese, or... Let me check.
Gotta love the power of Google.
Yeah. Hold on, let me check on this. Chlorophyll... What's it?
Oh, the Calvin cycle. I haven't, God, I forget.
Yeah, so it's magnesium. Alright, so you got a magnesium ion in the middle of this thing, and pretty much what happens is when light hits that molecule, it changes shape. And that shape change is coupled to essentially a conformational change that's mediated by electron transport through the chlorophyll tail. You get this cascade, and that conformational change in a protein via light absorption starts the cascade that allows you to turn light energy into physical energy by making ATP.
Nice.
Yeah.
And on the flip side to that is you make NADPH2, and that is used to transfer, basically carbon fixation.
Yeah,
so
carbon fixation.
You go 3 molecules of CO2, you go to 3, what's it, PGA?
PGA.
PGA. I forget what PGA means. Jeez.
Shout out to plant biologists.
Yeah, shout out to you guys. You guys are the true MVPs. Good Lord, I am so not up to date on this. Phosphoglyceric acid.
Yeah, there you go. So you go from
and then you go from there.
unstable 6-carbon intermediate to a 3-carbon PGA intermediate, and then you go to this 3-carbon. It's part of the Calvin cycle.
You make a bunch of 3-carbon sugars, and they add them all together to make glucose, and then you use the glucose in the mitochondria to make ATP.
Right.
And apparently, the Calvin cycle is much more efficient than the mitochondrial cycle.
It's all about
so that you make more ATP. That's interesting. I like that. Yeah, what are you gonna do?
What are you gonna do?
I don't know. I'm not gonna ever study plant biology, but shout out to you guys who do. Thank you. I really appreciate it because I'm more into the heterotrophic organisms. I think.
Let's get to the human stuff.
All right. Let's do one more thing. Multivesicular endosomes, lysosomes, etc. We had already talked about lysosomes being the garbage can of the cell, basically, and they
pretty much
break everything down.
They're the stomach of the cell.
Yeah. I like that. And they break it down to simpler molecules, and so these vesicles can be used as a transport system. So you go from one end of the cell to the other end of the cell. You can bring in things from outside of the cell. You bring them to mostly multivesicular bodies or multivesicular endosomes, and that's how you actually spit out exosomes.
Yeah.
It's pretty cool.
Exosomes are essentially vesicles that the cell uses to communicate. And I don't really know a lot about exosomes.
40 nanometers to 120 nanometers in length.
Yeah, in size and diameter.
Mm-hmm. I'm gonna study exosomes.
Yeah, pretty much they're big in cancer research because if you have a tumor cell that's transformed, it could spit out an exosome that can transform a neighboring cell.
Or it can actually be used as the seeders of a metastatic lesion. So
there we go.
Think of them as developing an area. So when you're building houses, you're going to have to lay in the groundwork, clear out all the trees. And that's what the exosomes basically do. They go and make the environment better suited for cancer cells that are going to later, slosh off of
and we call this
primary tumor
modulation of the tumor microenvironment. Exosomes are one of the mechanisms in which you can modulate the tumor microenvironment, especially by signaling to fibroblasts and creating what we call tumor-specific CAFs. Cancer-associated fibroblasts.
Yeah.
CAFs and TAFs. Tumor-associated and cancer-associated fibroblasts.
CAFs and TAFs. And another thing, you have endosomes that can deliver, like we said, bring stuff in from the outside environment of the cell into the inside interior portion of the cell, get that to where it needs to go.
Well, 3rd grade term, endocytosis. There you go.
Endocytosis.
That was the first science word I ever learned.
Nice. That's pretty cool.
When I don't understand something, I use photosynthesis to make myself sound more intelligent.
All right, so here's a review slide. We already covered all these things. I know this probably isn't our most well-rounded show. I know it's a very easy, very topical type of look at different things, but
it's interesting. You don't. The higher up you get, the less you know almost. It's like you know more about one thing, and the other stuff kind of fuzzes away a little bit.
Yeah. Well, I remember seeing.
If you don't use it, you lose it.
That is true.
Yeah.
40-year-old virgin. Is it true? If you don't use it, you lose it. But I remember seeing on the door of one of my undergraduate professors, and it was like a track through a PhD. It was like things you know about everything to the very fine point of what you do actually know. So you become a specialist.
Yeah, you become an expert. Yeah. Essentially, you just, you have to know all the background so that, you're well-rounded and so you can even start to try to understand the stuff that's super complex. There was a time when I knew plant biology, far, far away in the distant past. Anyhow.
I actually knew the Calvin cycle and what it did.
Yeah, right.
And what was involved in it, the enzymes and everything. All right, so let's talk about shit that I do know. Cell adhesion. I know this stuff. But all right, so you have adherens junctions, which is your classical cadherins. They attach to actin filaments. These are cell-to-cell contacts. Desmosomes with desmoglein and desmocolin. They are attached to intermediate filaments on the inside of the cell.
Which are structural proteins.
Yeah, the actin filaments basically made from actin. Intermediate filaments are made from keratins and different things like that. And
filaments are long polymers of protein subunits.
And they give support structure to the cells. In particular, extracellular matrix connections. You have actin-linked cell matrix junction, or integrins, which are sex